for. Okay, so I think last time we discussed, okay, until the carbohydrates, right? Yeah, the carbohydrates. Yeah, this one, okay, carbohydrates. And then the fats, okay, the fats. So whenever you study this biological molecule, it is very important for you to differentiate, okay, whether which one is monomer and then which one is a polymer. So for more detail, right, actually later I will give this to you, okay? I'll give this to you. Where is it? Okay, you can just refer to the sentence, okay? This one. You should know what is monomer, what is polymer. Monomer is just they refer to the one units. Okay? If you link those units together, you become a large complex structures, which is a polymer. So whenever we talk about the monomer, we are actually referring to the glucose, the fructose. So you join a lot of glucose together. When you link them together, it will become a large complex molecule, which is a polymer. So it will belong to the starch, glycogens, and the cellulose. So those are made from the glucose molecule. It's made from about thousands to hundreds units of the glucose molecule. And then maybe monosaccharide, okay, for this one, monomer, glucose and fructose. Is it soluble in water? What do you think? Is it soluble in water?
Yes.
Yes, and then how about why is soluble in water? Why those monomer, glucose and the fructose, they can they're able to dissolve themselves in water, is because their site is very small. And then the glucose and fructose, right, they have their hydrophilic parts. In the glucose, when you when you think back their structures, right, the glucose, they have the OH, the hydroxyl group, the OH. So this OH group, they can form the hydrogen bond with the water. So that's why they can dissolve in water. It's a soluble molecule. But when you have a lot of thousands of the glucose unit joined together, you become the polymer, which is the starch, the glycogen, and cellulose. So now they'll become the polysaccharide, and it is not soluble in water. Okay, Neo. So why polysaccharide is not soluble in water? Why it is insoluble in water for the polysaccharide?
Like the chain is too big for for water to dissolve it, break the chains. Like the molecule.
Yes. Correct. It's too big for them. Okay, it's too big for them. Yeah. So for the monomer, right? Yeah, it's amino acids. So you have about hundreds unit amino acid joined together, you become the polypeptide chain. So later the polypeptide chain, they will fold together, become the proteins. Okay, so later this one I send to you. Okay, you can just refer to the English part. Okay, you ignore the Mandarin. Sorry, you ignore the Mandarin. Okay, yeah. So this is a protein. You have a lot of the amino acids okay, the amino acid, they join together, become a peptide. Okay, so the peptide, they can fold to each other and then become a final product, which is a proteins. And then today we're going to talk about the lipid. I think we talked about lipid before, so yeah. So for the fats, okay, fats, or sometimes we call as the oil and the fats. Okay, fats is actually, they refer to the solid at the room temperature, and the oils type, right, they are actually referring to the liquid at the room temperature, and they are referring to the triglyceride. So triglyceride itself, it is a polymer. Triglyceride is a polymer because triglyceride is made from the monomer, which is from the 1 glycerol and 3 fatty acids. Okay, it's made from monomer, which is those two structures. Yeah, okay, just 1 glycerol and there's 3 fatty acids. Okay, Neo, can lipid dissolve in water? Can lipid dissolve in water?
No.
No? Okay. It's because of what? It's because that the lipid, right, which is triglyceride, they are unable to dissolve in water because lipid itself is non-polar. What's meant by non-polar? This one, non-polar. Water is a polar molecule. So you just remember in this way, okay? Polar molecule only can dissolve with polar. So that's why the non-polar molecule, right, they are unable to form the hydrogen bond with the polar. So the non-polar is because the triglyceride, they have a very long chain of fatty acid too. So you can see, right, this is a fatty acid too, and then it's very long chain like this, okay? Those are the long chain of the fatty acid too. It comprised of the hydrocarbons, which is a hydrocarbons. Hydrocarbon. Because you can see the chain. This is a long straight chain. It's just made from the carbons and the hydrogens. So they call it hydrocarbons. Hydrocarbons. Okay, like this. So once you join the one glycerol with 3 fatty acids, it will become 1 unit of triglyceride. And then this process, right, you produce about water. Okay, you produce about 3 water molecule. You can add here 3 water molecule. Which is the one glycerol, they can join with 3 fatty acids by bond. Okay, so where's the bond? The bond, the name it called is what? Ester bond. So how they can form this is actually they are removing the 3 water molecule away. So they are actually removed here. Oh, sorry. Smaller. Okay. Here they actually remove here to here. Okay, so that's why H plus OH become H2O. So by this way, they can form about 3 water molecule. So where is the ester bond? Ester bond is at here. Here is the ester bond. Okay, here. So it's a bond joining between the fatty acids and the glycerol. Yeah. So this process, right, it also called as what? It's called as condensations or hydrolysis. Maybe you tell me this process, right, is known as condensation or hydrolysis. Which one? You combine the 1 glycerol and 3 fatty acid and then become triglyceride. So you think it's condensation or it's hydrolysis?
Maybe condensation? Wait, is it condensation that like makes more or?
Yes, the answer is condensation. Yeah. Oh, what happened to mine? Okay, yeah, it's condensation. Okay, just remember that combine. Okay, just like last time we learned about when you combine 2 glucose molecule become 1 unit of maltose. That's by condensations. Condensation means combine. Okay, means combine. Combine 2 molecule become 1 complete units. So this condensation, right? And then what they will do is there's another name. Okay, for this the forming the lipid molecule, there's another name. We call it what? Esterification. Some of the book they will mention this up. Esterifications. Because you're actually forming the ester bond, joining these 2 molecules together, become 1 large molecule. So this is condensation. Okay. So every time once the biological molecule they do the condensations, they will produce about water molecule. Okay. So how they can do the condensations? It's actually they are removing the water molecule away, so they can form a bond joining them together. So this is applied to all kinds of biological molecule, no matter it's carbohydrates, proteins, or lipid. Okay, so later we can have a look. Yeah. So triglyceride, okay. Okay, for the lipid, right, we learn about 2 types of lipid. One it calls triglyceride. Tri means 3. They have about 3 fatty acids, so they got triglyceride. Then how about another one? We call it this one, phospholipid bilayer. So this phospholipid bilayer, so these structures, so they only have about 1 hydrophilic head, right? So this 1 phosphate head, because they contains the phosphorus atom. So you can see like these P atoms are. So this is phosphorus. So the phosphorus, they are in a charge because phosphate ions. Okay, they contain 1 phosphate head, and then they have about 2 fatty acid. Okay, 2 fatty acid, 2 fatty acid. Yeah, so you can see, right, this is this structure like this, right? Like this. Actually, it's referred to the hydrocarbon chain. It refers to the structure like this. Okay, it refers to this one. The same thing. Just like this is the structure formula, and then just now the structures is we call it skeletal formula. Because you will learn chemistry in another class, right? So that's why this is how they draw the structures. Sometimes they will draw in this way, okay, like this. Like a zigzag shape. So this zigzag shape, right, is actually they refer to this hydrocarbon chain. It's the same thing, just different presentation. So like this, right? So the fatty acid is composed of a long hydrocarbon chain, and they will have a bonds. Okay, sometimes they will have we call it double bond. This one, like this. So this is the double bond. So if the fatty acid itself, they will have a double bond, right? They call as what? They will have a unsaturated fatty acids, okay? They'll have a unsaturated fatty acids. And then if you have single bond, only single bond, there's no presence of double bond, we call as what? We call as saturated fatty acid. Okay, so because of the presence of unsaturated fatty acids, so that's why the tail part of the phospholipid molecule, the tail part of the phospholipid molecule, they'll form a king like this. Just like your leg is slightly raised up. Okay, so this structure look like this. And then now we talk about proteins, right? Proteins. Okay, protein is made from what? It's made from the amino acid like this, okay? So this is amino acids. And then in our body, right, we have about 20 different type of amino acids. So these 20 different type of amino acids, right, they can join in different sequence. So this different sequence, they'll form the different structures of the polypeptide change, okay? So this amino acid, right, they can join, okay, because you have 20 type. You have about 20 type. So it can join to become a different sequence of amino acids. Sequence of the amino acids. And then this different sequence amino acids, right, later they will become what? Later they will become the different structures, okay? Different structures of the polypeptide chain. Proteins, okay? And then this different structure of protein, later they will have a different function of protein. So for example, what is the example of protein product that you learned before? You learn about enzyme, okay? You learn about enzyme. You learn about antibody, the hormones. Those are the protein products. So every different kind of protein product, they have a difference in their structures and the shape due to the different sequence of amino acids. Just like this way. And then how the amino acid look like? They just look like this, okay? So you need to remember about these structures. Whenever you want to draw the amino acid, right, just draw in this way, like this C, and then C, OOH, okay? Amino acid, they have a group we call as carboxyl group. This is a carboxyl group. Carboxyl, okay, this one, carboxyl group. It's belong to the acid, okay? It's belong to acids. And then you have about side chain R, and then you have about hydrogen atoms, and then you have about a minor group, okay, which is NH2. Yeah, so just appear in this way. And then for the amino acid, right, they will have what? They will have this one, okay, COOH. What is COOH? COOH is carboxyl group. Okay, it's an acid. So that's why it's acids. Acid, they can donate the hydrogens. Because the role of the acid is they want to donate the hydrogen atoms. Okay, so that's why hydrogen can be go out. Then how about the base? Base is the NH2. This is the base, okay, NH2. Base. So what the base will do? Base, they will tend to accept the hydrogen atoms. So they go to here. Okay, so at the end, you form this kind of charge. Okay, because COOH, right, they lose one hydrogen atom, so that's why they become negative. And then the NH2, which is the amino groups, they will accept one hydrogen atom, so they will become like this, NH3 plus. Okay, so that's it. So this is just showing that amino acids are in their ionized form because they have a charge, okay? Ionized form, the amino acids. Amino acid. Okay, so that's why you have about 20 type of the amino acid, right? So each of the amino acid, they have a same structure, which is they have a carboxyl groups, the base. So they just have same structure like this. Okay, so that's why amino acid, they can be arranged in any order since you have about 20 type. So you resulting about different kind of protein product being produced. Okay, so maybe you can think about like in this way. So this is amino acids. So how they can join together to form a dipeptide. So later they become a polypeptide. So it's actually there'll be two monomer. Here is a two monomer. Here and here. Okay, you have about two monomer, the amino acids. Okay, they just join together, right? They'll become a dipeptide. So here is a dipeptide, okay? So here is here is the bond, okay, of the dipeptide like this. Because what? Peptide bond. Just like in the lipid, you have the ester bond. So in the protein, right, you have peptide bonds. So this peptide bond, they are just linking the amino acid together. So you can see it's condensation. So because they are actually removing the water molecule away. So that's why condensation always removing of the one or two or more water molecule away. Become like this dipeptide. So once the body, okay, they want to break down the dipeptides, right? So what they can do, they can add back the water. See, they're just adding the water back. So later, so this dipeptide, right, they will spill. Okay, they can spill, become each of the amino acids compound. So this is called what? Hydrolysis. It's because they are actually using the water. They're using the water to break down the molecule. So that's why they call it hydrolysis. And a lot of our digestion process actually using the hydrolysis to break down the food. Okay? Do you realize that a lot of the digestion process, right, which has happened in our intestines, they're actually using this hydrolysis to break down the food. Okay, maybe I can give you example. Starch, okay, maybe. Last time we talked about starch, right? Starch. So starch is a monosaccharide, disaccharide, or polysaccharide. Which one?
Polysaccharide.
Yeah, starch is a polysaccharide. So now you're going to break down the polysaccharide. So what kind of enzyme used to break down the starch? What kind of enzyme?
Amylase?
Yeah, it's amylase. Yeah. Okay, you are so smart. Yeah, amylase. So that's why starch. And then they'll become what? Later?
Glucose?
Maltose. Maltose first.
Maltose?
Yeah, it's maltose first, yeah. Yeah, so that's why you have a what? Later you have a food test, right? The starch, right? The starch, okay, because what? Iodine test, yeah, you have the iodine test. Because initially, the iodine is, what is the color of the iodine naturally? The antiseptic solution, the iodine. The natural color of iodine is what color?
Like purple, like blue, dark
Brown,
blue.
usually it's in brown
Or
color.
brown.
Yeah. So see, this is the iodine, okay? It's brown in color. Actually, iodine is referred to the antiseptic solution that you apply. This one, okay, iodine solution. Whenever you get injured, you will apply this iodine. This one, this is iodine. See? Yeah, the iodine solutions.
Okay.
Yeah. And then, so that's why you can do a test, okay? You just put the iodines on the food which contains starch. For example, rice, biscuit. And then you see the color will change. So the color will change to become what? So what's the color? Dark blue. Here. From brown color, right, they will become dark blue. Because there is a presence of starch. Due to the presence of starch. So that's why, the starch, okay, plus water and then break down by amylase become the maltose. Yeah. The end. So that's why this is we call as what? Hydrolysis. Okay, this solution, this process, right, we call as hydrolysis. Because you are actually using the water to break down the molecule. Same to the protein, okay? For example, the proteins, okay? So every time when the food particle, they need to undergo the digestion process, right? They require the addition of water because they need the water to break down their bond. So protein plus the water. So tell me, what kind of enzyme will break down the proteins?
Protease?
Enzyme break down the protein? Protease, right? Protease.
Yeah, protease.
Yeah, protease. This one. I think you learned about few proteins before, which is the trypsins and pepsins. Pepsin in the stomach and trypsins in the in our small intestines. So that's why they can break down this long chain of proteins become dipeptide. So maybe from in this way, right? Okay. Initially the long chain, right? They break down. So at the end they become like this. Which is called dipeptide. Dipeptide means that they have about 2 amino acids linked together. So this is called hydrolysis. Hydrolyze means breaking down. So they will involve in the food digestion process. Okay. And then okay, maybe you can think about how the monomer, which is the amino acids, they able to become a large and complex protein structures. So that's why the proteins, right, they are being classified at the 4 level of organizations of proteins. So from the primary levels to secondary, tertiary, and the quaternary, which is the most complex form of protein. So initially, right, in the primary structures, they will have the amino acids. So this amino acid, they will join together, okay, like in chain like this, okay? So this is amino acid. So they are connected by the peptide bond. Here's a peptide bond, okay? And then they will have the beta pleat sheet. This is the beta pleat sheet. Okay, Neo, how they can able to form a beta pleat sheet? Just imagine that this is a paper, right? Okay, they can just like roll down themselves like in this way, okay? Let me show you, okay? This is a chain. This is a chain, okay, like this. Okay, a straight chain. Where's my computer? Oh, yeah, okay. Like this in the straight chain, right? When they roll down, okay, like this, okay, they just roll down. They just coil like this structures. So now they become what? Secondary structure. Okay, so it's in the beta pleat sheet. So this is a protein in the second structure. They have some, they have some structure already. And then now, when they move into the tertiary structure, right, they just fold them together like this way. So now it requires what? Globular shape. Okay, so Neil, what is meant by globular shape? This is a globular shape, which is circle, round in their shape. Okay, so this is referred to the tertiary structures, which is a polypeptide chain they fold due to the interactions bond between the amino acid group. So later for the tertiary structure, right, if you have another proteins, okay, another papers, another protein they join together, right, for example, like in this way, okay, so now they'll become about a few polypeptide chains joined together. You have a 1 again, and then 2 joined together, become like this, right? So now they become what? Quaternary structures. Okay, so Neil, do you understand about the process?
Yeah.
Yeah, okay. Okay, so that's why tertiary structures consist about 1 or more than 1 polypeptide chain. Tertiary structure, you have about 1 polypeptide chain or you have more than 1 polypeptide chain. Which one? What do you think?
Wait, could you repeat the question?
Okay, the tertiary structures, right, they have about 1 polypeptide chain or they have more than 1 polypeptide chain. Only 1 chain or they have more than 1 chain, maybe 2 or 3 chain. What do you think?
More than one chain.
Only 1 chain, yeah, correct. Only 1 chain. As you see in this picture, right, there's only 1 chain. Which is that chain, right? They just fold themselves, become a global structure. So this is tertiary structures. Then how about in the quaternary structures? In the quaternary structures, they will have about more than 1 chain. So like in this picture, right, they're showing about 4 chains, okay? You have about 1 polypeptide chains, seconds, thirds, and fourth. So in the total, you have about 4 chains. 4 polypeptide chains joined together. 4 polypeptide chains joined together. Yeah. And they will become a quaternary structures. So this is a final complex. Final structure of the proteins. Okay, so for the protein itself, the shape and the structure is very important. Why? Why the protein shape is very important? It's because the shape of proteins will determine their functions. Do you agree with me?
Yeah.
Okay, because when the proteins you look at the actual ways, right? Let me show the actual protein structures. They are like in this way. Okay, where is it? Okay, here is it. This is the actual enzyme structures. Okay, can you see it? There's a lot of change. The polypeptide chain, okay, they just fold in this way, yeah. So they'll form a final complex form, which is the enzyme. Okay, so it's very, very complex in their structures. So like this, okay, very complex in their structures. So this is the actual protein structures. So that's why the structure of proteins can determine the function of protein, because every kind of the protein products, right, they have their own unique shape. Okay. Different type of a protein product, they have their own unique shape. Just imagine this way. Okay, for example, right, the proteins, okay, which is this one. The proteins. Okay, example of protein product shown in this picture, right? You have the red blood cell. So this is a red blood cell. So the red blood cell, they will have what? They will have about they're made from the hemoglobin molecule. So that one hemoglobin molecule, just that one hemoglobin, hemo means red blood cell. Globin means proteins. So this hemoglobin, they are made from 4 chains, okay, which is the this one beta chain. Okay, this is beta chain and 2 alpha chains. So this alpha and beta, right, they are just referring to the secondary structure of proteins, which is just now, okay, they just like helix, okay, we call it what? The helical shape, okay, the helical shape are like this. So this is structures for the hemoglobin. Then how about in other kind of the protein product, which is the antibody? So you can see here is an antibody like this. Antibody, 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 okay? So every antibody, right, I mean each of the antibody, they have their unique shape, which is like here. So you can see, right, this is different compared to here. And compared to here, it's different. Because each of the antibody, they have their difference in their, this is, we call it what? Different in their structures for the antigen binding site. So this is the antigen binding site, which is the site on the antibody. They were able to bind the antigens on the pathogen surface. Antigens binding site. So whenever this bacteria, right, okay, this is from the pathogen, okay, no matter it's bacteria or virus. So every pathogen, right, they will carry their own antigen. So this is the antigen, okay, the yellow color, right, is referred to the antigens. Antigens, okay, on the pathogen surface. Pathogen surface. So that's why the antigens, they need to bind to the antigens binding sites on the antibody. So at the end, they'll form what? They'll form the antigens antibody complex. Okay, because you have about different type of pathogen, right? So that's why they need to be killed by different type of the antibody. And then so this is showing the enzyme. Okay, every enzyme, each enzyme, okay, each enzyme has their own unique shape of the active site. So which allow the substrate, okay, substrate with the complementary shape to bind with it. Okay, which is every enzyme, they have difference. I mean, they are actually different in their structures for the active site. Yeah, which is they refer to here. Okay, the active site structure is different for each of the enzyme. So that's why they can only allow the certain, I mean, they can only allow the specific type of substrates they can bind to it with a complementary shape. So if the substrate itself, their shape, right, is not complementary to the active site of enzyme, so it means the substrate unable to bind to enzyme. So they cannot form the enzyme substrate complex, and then they will not produce a product. So that's why this is the shape of the proteins, because it will determine functions. Okay, so that's why every proteins, they has their own unique 3-dimensional shapes. So they can enable them to carry out their functions. Okay, that's all. So we finish our proteins, and then now we talk about food tests. Okay, food tests, you just need to know about, there's few important food the food tests. So the first one, we talk about the Benedict solutions. So this is a Benedict solution, right? Benedict solutions is initially blue in color, like this way. See, this is blue in color. This is the original color for the Benedict solution. So this blue color solutions, what they will do is they will change to become a green color, red, orange color until become red color, if there is a presence of the reducing sugar. They only change color if there's reducing sugar. I mean, they will only change color if there's a reducing sugars presence inside the solutions. And then you remember in the Benedict solution test, right? They require heating. Okay, you need to make a water bath and then to heat the solution up to 70% for about 5 minutes, and then you can observe the color change. from the blue to the brick red precipitate due to the presence of the reducing sugar. So what's actually referring to reducing sugars is referring to the sugar, right? They will have the free groups, and then they can do the, we call it the oxidations or the reactions with the Benedict solution itself. So this is example of reducing sugar, okay, which is glucose, fructose, lactose, maltose, galactose. Actually, all of the monosaccharide and the disaccharide, they are reducing sugar, just except one disaccharide, which is sucrose. Okay, sucrose is not reducing sugar. So that's why when you mix a sucrose with a Benedict solution, right, the Benedict solution, they will still remain in blue color. They will not change to brick red precipitate. But when you mix a reducing sugar with the Benedict solutions, the Benedict solution, they will change color, okay, from blue to brick red precipitate. So just remember that all monosaccharide, okay, and the disaccharide, just accept this sucrose. Accept the sucrose. Is belong to what? Is belong to the reducing sugar. Okay, for the polysaccharide like the starch, glycogens, and sours, they are definitely not a reducing sugar. Okay, they are also belong to the non-reducing sugar. So that's why I can add more thing, which is the starch. Okay, the starch, the cellulose, then the glycogens. Yeah. So this is the experiment setting. Okay, you put the food samples together with the test indicators under the water bath and burns with the Bunsen burners about 5 minutes, and then you can see the color change. Then next, we're moving to the protein test. So how we're able to detect there is a proteins inside the food sample. So you use this, we call it what? Biuret test. Okay, so this is a Biuret test. Biuret test, right? The color also is blue initially. But if there's a presence of proteins inside the food sample, the Biuret test will change from blue color become purple color. So if the solution just remain blue color, right? This means that the food sample itself, they does not contain proteins, because they will only change colors to purple color if they have a protein presence inside the food sample. And then now we talk about iodines. Okay, so this iodine is the most commonly asked questions in the exam. So just remember iodines, right? The initially color is brown. Okay, it's brown, and then they only change their color become dark blue if there's a starch present inside. So that's why initially the starch is present, but after that, when the starch is break down by the amylase, they will become what? Maltose. Okay, so this is maltose. It's a disaccharide. It's a maltose. So this maltose molecule, what they will do is they will cause the iodine solutions. Can they change in color? Okay, so now my question is, can the iodine solution change in color if there's presence of maltose molecule? There's no starch already, because all starch become maltose. Can the iodine solution still change their color, or they just remain as their brown color?
They all change color.
Yeah, they will not change in color because there's no starch inside already. Because all starch is being break down become the maltose. Yeah, okay, so here is a test that you can try for it. We call it as what? Iodine test, okay, the iodine test. So now we talk about the lipid, okay? So this is whether how we're able to test the food sample, they contain the lipid or not. So they will use this, we call it what? Emulsion test, okay? So this is, we call it emulsion test, to test for the presence of lipid in the food sample. So you can, okay, so you can have a look, okay? This test tube, right? This test tube, they will have what? They will have the ethanol inside. Okay, they have a food sample and the ethanol. And then another one is they will only have the distilled water. Okay, so what happened is you just mix the food sample with the ethanol. So if the food sample, they consist of lipid, right? So this lipid, which is a non-polar, they can dissolve with the ethanol. Ethanol, they have a non-polar portion. So that's why these lipids, which is non-polar, they can dissolve with the ethanol, which is also non-polar. So later they will create a positive result, like this. So they will form these cloudy emulsions. Okay, cloudy emulsions. Like this. Okay. So this shows that there is a lipid contents inside the food sample. So if the lipid are present, a milky emulsion will form. So the solution itself will become more cloudy, not transparent at all. So the more the lipid present inside the food sample, the more obvious the milky color of the solutions. So this is called what? Emulsion test, okay? Emulsion test for the lipid. You need to mix a food sample with the ethanol because ethanol is a non-polar. And then you add the water, okay? Water to the ethanol itself because you want to dilute the solution. And then you form the emulsions, okay? The cloudy emulsions. So this one you call as what? Cloudy emulsion, okay? Cloudy emulsion. Then how about the vitamin C test, okay? If you want to test for the presence of vitamin C inside the food contents, you should buy these DCPIP solutions, which is initially also blue in color, okay? Also blue in color, same to the Biuret test and the Benedict solutions, which is initially blue in color. So that's why what the DCPIP would do is initially it's blue in color, right? But when it's being added with the food sample, consists of the vitamin C. So from the blue color, they will change become what? Colorless. From blue become colors. So that's why they can change their color, become transparent form. Okay, so that's why the positive test will show that when the blue color of the dye disappearing. So this is a vitamin C test, okay? If they remain as a blue color, right, that means this food sample, they do not have the vitamin C content inside. Okay, we all finished about the food sample, okay? So just later you can read back the here, okay? This one. Okay, so that's why we talk about the carbohydrates, proteins, and the lipid. And then you should know about the bond, the monomer and the bond, okay? So the carbohydrate, right, actually is formed by the glycosidic bond, which is between two monosaccharide, peptide bond and the ester bond, okay? So later we'll talk about nucleic acids, just give me a few minutes. Okay, so here's a test, okay? Here's a test for the proteins, lipid, and carbohydrates. Okay, and then now we talk about what? We talk about nucleic acids. Nucleic acid, okay? Nucleic acid is referring to what kind of structures? What kind of structure that you learned before is classified under nucleic acids? Do you remember? Nucleic acids. The DNA and RNA.
Yeah.
Yeah, okay, the DNA, RNA, they are under the nucleic acids. So the DNA, RNA, okay, your DNA or RNA is polymer or is monomer? You think it's polymer or it's monomer?
Polymer?
Yeah, it's a polymer, right? Because we talk about the DNA, right? Oh, the DNA is considered a very long chain double helix, like this, okay? So this is the DNA, okay? It's a very long chain and it's double helix because they made from two strands. They just join together. So that's why it's a polymer. And then the polymer DNA, right, is made from the unit, which is called as what? Nucleotide, okay? So this is a nucleotide. So this is a monomer for the DNA. So nucleotide, right, they have about 3 components. First, they will have this, we call it as a nitrogenous base, made from the ATCG, okay? The nitrogenous base. Nitrogenous base, made from the ATCG. And then you have about a pentose sugar, which is a 5-carbon sugars. So this one is pentose sugar, okay? We call it what? Deoxyribose sugars. So this deoxyribose sugar is in the DNA, so that's why it calls deoxy. So this is a pentose sugar, it's 5-carbon ring, right? And then you are connected to the phosphate group. So that's why this is the structure of nucleotide, okay? You have the base, sugar, and the phosphate group. So they will form the backbone of the DNA. Okay, so that's why when you join the nucleic acid together, right, it becomes like this. Okay, so each of the nucleic acid, they just join to opposite strands by the bond. In the DNA or in the nucleic acid, you should know there's 2 bonds very important to maintain the structure of the DNA. The first will be the hydrogen bonds. You can see, right, here is a hydrogen bonds. Hydrogen bond, okay, which is the bonds is forming connections with the hydrogen atom. So that's why they call it hydrogen bond. So between the the base pairings of the DNAs, they will have the hydrogen bonds. So the A and T, right, actually there's 2 bonds. However, in the C and G, right, they have about 3 bonds, okay? So this just for extra knowledge already. So that's why A pair with T, C pair with G. It calls ATCG. So this is called as what? This is called as a complementary base pairing. This one. Complementary base pairing. So you always have A to T, C to G. And then another bond called as what? This one. Sugar phosphate backbone. So this is a bond which is connecting from one nucleotide to another nucleotide, which is their adjacent part of nucleotide. So this is called as what? Some of the book, right, they call this a sugar phosphate bond. Sugar phosphate bond. Some of the book they call it as a phosphodiester bond. You can choose either one. Phosphor. Yeah. So this is from the strong backbone of the DNA. Okay, so in actual way, right, they just look like this. Here is it. So you can see, right, here is the hydrogen bond. Hydrogen bond. And then here is the sugar phosphate bond. Here. Which is they're actually joining from one nucleotide to another nucleotide, which is this adjacent part only. It's not opposite that, but it's adjacent part. So this is called sugar phosphate bond here. Okay, there's a sugar phosphate bond. So you can see this is the structure of DNA. Okay, they form from double helix. Okay, Neo, DNA has 2 strands. How about RNA? RNA also has 2 strands, or they only have single strand. RNA. What's your answer? RNA, what do you think? RNA have 2 strands like DNA, or they only have single strand?
A single strand.
Yeah, RNA, they have a single strand. Maybe you're wondering, right, what's the difference in the DNA and the RNA structures? The difference is DNA double strands, RNA is single strand. Another thing is, when you look at DNA structure, right, you can see like in these pictures, DNA is just a very large and complex molecule. No ending, look like no ending, because it's very, very long, super, super long. So that's why it's very hard for the DNA to go out from the nucleus to the cytoplasm. It's very hard for them because they are too big. And then inside the DNA, they have what? Inside the DNA, they have a genetic information. Because the function of DNA is they just want to store the genetic information in form of gene. So that's why the genetic informations will be used up by the body cell to make the proteins or any kind of the products. So that's why the DNA, what they do is they will uncoil. Okay, so you can see this is portion that the DNA just unwind themselves. Like this, from like this, right? They're just like this. Okay, unwind themselves. So why they need to unwind themselves or uncoil themselves? This one. First, DNA will unwind or uncoil. Okay, because they want to synthesis the messenger RNA. Okay, so this is a mRNA, we call it messenger RNA, okay, from the gene located on the DNA strand. So that's why between the RNA and the DNA, they can form a base pairing. Another thing is the difference is the DNA, right? They have what? DNA has 2 strands, RNA have single strand. And then DNA, they have this one, ATCG, okay? DNA, they have about 4 base. ATCG. How about in the RNA? They also have 4 base, okay? But the A, right, is being replaced with U. C and G remain the same. Because in the RNA, there's no thymine, no T inside the RNA. So that's why whenever they want to form the base pairing, you should pair up in this way, okay? Like the A, okay, see A from the DNA, right? They can pair with U. C pair with G. Just A to U, A to U. And then T to A. Okay, so this is happened during the transcriptions. So the purpose of transcription is they just want to produce a messenger RNA, and then this messenger RNA is much more shorter, lighter, so they can carry that specific genetic information from the DNA out. Okay, from the nucleus, right? See, they just travel out, reach the cytoplasm. So that's why the mRNA, they can bind to the ribosome. mRNA, they just bind to the ribosome. So the ribosome, they just move along the mRNA, and later they can read through the informations. So what the ribosome will do, ribosome, right, they will do the assemble the amino acid together. Ribosome, they will assemble amino acid together to become a polypeptide chain. Yeah, so this is how the proteins are able to produce. Okay, initially they just start off from the genetic informations in the nucleus. Okay, they'll just give out the mRNA. Okay, mRNA, right, just like provide the guidelines. So you just imagine like this way. Okay, this ribosome, they just read on the guidelines how to make proteins. Because ribosome, they'll prepare the materials. So ribosome, right, they can allow the formation of the peptide bonds occurs in their sites. So that's why ribosome, they can help to synthesis proteins. So, if the question asks you about function of ribosome, this is what you should write. Ribosome synthesis proteins. Okay, because ribosome is the one who can allow the peptide bonds to join between the amino acids, and then they will become a polypeptide. So this is all happen in the ribosome. So ribosome, they'll carry out what? They carry out the translations. Because this translation refer to what? Refer to the translation of proteins. They are referring to the genetic informations carried by the messenger RNA, right? They can be read by the ribosome, so the ribosome can transform that, become the actual protein product. So this is called translations. Just now the transcription refer to what? Just now the transcription refer to from the DNA, right? From the nucleus, DNA in the nucleus, become the messenger RNA. Okay, so Neil, are you clear about it? Yeah. So that's why this is, that DNA, the nucleic acid and protein is different. Two different things. Don't think that the DNA can use to make protein. Cannot. Protein is made from the amino acids, which is a protein. But DNA, they only can provide the information to make the proteins. Maybe I can ask you this way, okay? Can the proteins being produced without the information from the DNA? If there's no information from DNA, right, can the protein still able to do the synthesizing process? Yes or no?
No.
No. Yeah, no. It's because there's no information from the DNA. So like this is a gene, okay? The gene. Okay, what's meant by genes? You should able to differentiate gene and the DNA is different thing. Okay, DNA is like this, okay? It's a whole strand, very, very long. No ending, okay? You cannot see ending. This is DNA. Okay, so the gene, right, it just refer to the specific regions, a length, just a specific length, a short length of the DNA. Okay, so this is gene. So that's why the gene, right, in another words, they call as what? A length of DNA, okay? A length of DNA which encodes for proteins. So this is the gene. So later the genes inside the DNA, right? Gene is located inside the DNA. They can transcriptions to produce a messenger RNA to provide the guidelines to do the protein synthesis. So this is called transcriptions, which is the information from the DNA, right? They are coded into the genes and transcribed. Okay, see, they are transcribed in the messenger RNA. So later the messenger RNA, they'll go out, okay, and then they'll bind to the ribosome, do the translations. So that's why the coded information now in the messenger RNA, right, is translated to the protein. So this is how you get the protein, the polypeptide change. Okay, I think currently is like this. Maybe you can try some question. Okay, I think we almost finished about chapter 4. Yeah, chapter 4. Okay, water, right? Water. You need to know what's the importance of waters in our living organisms. So the water, right? Okay, water is made from what? Water is made from the hydrogens and the oxygen molecule. Water equals what? H2O, right? H2O, you have about 2 hydrogen atoms and 1 oxygen molecule. And then water is a polar. So why water is a polar? So can you tell me why water is known as polar molecule? Why? It's because they have what kind of structures, or they have what kind of elements, so they're known as polar molecule? Your answer is? Because they have charge. Okay, water is, they have a charge. They have we call it electron negative charge and electron positive charge. So they have a charge on the oxygens, negative charge. They have a charge on the hydrogens, which is a positive charge. So that's why a lot of the ionic compound, right? Ionic compound, which is they will carry the charge on their molecule, like this showing that what sodium. Okay, see, this showing that the salt solutions made from the sodium and chloride, they can dissolve in water. So why the salt solution can dissolve in water? It's because the salt solution, they have this Na plus. So this Na plus, right, they carry the positive charge, so they can attract to another molecule, which is from the oxygens, right? Here's oxygens, so which carry the negative charge. Okay, so the positive to negative charge, they will form the bond, okay, the attractions. How about chloride? Okay, chloride is, they will have the negative charge, right? They have negative charge, so that's why they can bind with the hydrogens, right? Which is only have the positive charge, like this, okay? So you just remember in this way, because always, so this is natural process, which is the positive parts always attracted to the negative part. So that's why the salt able to dissolve in the water. Okay, so that's why this made the water become a very important solvent, because they can allow a lot of different substance, right? Just ensure it's polar. Okay, it's a polar. They have a charge on their content, so that's why it can be easily transported around the organisms. And also, the digested foods in the alimentary canal, which is your intestine tracts, right? They need to be moved to the cell all over the body. So that's why the water will help to transport the neutral molecule, like the oxygens, the carbon dioxide, the glucose, the amino acids. Yeah, so that's why water, they can help to transport the digested food. And then they can allow the food molecule, right, or the neutral molecule dissolve in the water. And then for this one, okay, water, right, they can help to remove away the toxic substance such as the urea. Or if your body have excess form of salt, if your body have excess salt, right, there's too much salt inside your body, so your body will respond, okay, by removing away the salts or the urea, by increasing the urinations. So they will increase amount of urinations, trying to flush the toxin out from your body. Okay, and also water also very important part of the cytoplasm, and they play role in ensuring the metabolic reactions can happen. Yeah, so that's why a lot of metabolic reactions, no matter it's your anabolic reactions or catabolic reactions, they require help from the water. Okay, just like we showed about what hydrolysis. Okay, hydrolysis also require the input of the water. Okay, that's it. So we come to the quiz sections. Okay, yeah, this is a question come out in this year exam. Okay, gene found on specific position of biological molecule. State the name of biological molecule. Which one? What's your answer? The 4 biological molecule that we learned, okay, which is the lipid, nucleic acids, proteins, carbohydrate. So you think this is referring to which biological molecule? What's your answer?
I don't really know.
Yes, sorry. Okay.
I don't really know. Look.
Okay. This is like this. Okay. Just so we talk about what DNA, right? The DNA. Like this. This is a DNA. Okay. This is a DNA. So this DNA is a very long strand, no ending, very complex. So where's the gene? Gene is just a short segment on the DNA, right? Just a short segment like this. Correct? This is the gene. The gene. Maybe the gene A, and then the gene B. So later, this gene A, right, they can use the information in the gene A to code for the proteins, which is protein A. And then this is gene B, right? They can use the information stored inside the gene B. Later, they'll undergo the transcriptions and translations, and then they'll become the protein B. So this showing that gene are found at specific positions on a biological molecule. So what kind of biological molecule you can discover gene? The answer is the DNA. Okay, correct, DNA. Okay, so Neo, do you understand this question? Because the DNA, right, is a very long, and then there's a portions, they have a gene inside. Okay, do you get me?
Yeah.
Maybe I can explain this way. Because Neo, you just imagine in this way, your DNA is very long, okay, very long and very complex, right? And then there's not every regions on the DNA they can use to make the proteins. Understand? The whole DNA region, right, cannot use to make the proteins. Only certain part, like in this part, in this part. So in this part, right, which is another part here. In this part, right, the genetic information is not used to make proteins. Because we call as what? Non-coding regions. Non-coding regions refer to the sequence nucleotide based on the DNA. They are not used to make protein later. And then you have a coding regions. Same region, coding region. Okay, so this one, right, some of the students, right, the answer, genes, they are found at the specific position on a biological molecule. The answer is proteins, okay? The answer protein, which is a wrong answer. The answer is DNA, okay? Or sometimes you can write nucleic acids as well. It's a DNA, because DNAs, they will discover the gene. Yeah, so the gene are found in the DNA. So the gene, right, they are not becoming the protein yet. They just found in the DNA. So later, they will have a short segment. They will become the protein later. And then for this one, okay, what are the individual units that are linked together to form a DNA polymer? What are the monomer? They can form the DNA polymer. Your answer is?
Nucleotides?
Yeah, nucleotide, correct. Then how about protein? It is an amino acid. Yeah, well done. So, we already finished talking about biological molecule. Just remember that biological molecule, right? They refer to the molecule. They consist of carbon and hydrogen. These two things. So that's why when you see the elements, so we can see sometimes the question asks about elements, right? They'll ask in this way, okay, state the elements in the carbohydrate in the protein. So this is the thing that you should write. So the carbohydrate and the lipid, they are the same. Okay, they have a carbon, hydrogen, oxygen, but the protein nucleus is different. In the proteins, right, the elements, it consists of what? In the proteins, they have a nitrogen and the sulfur. This is in the protein. They have nitrogen and sulfur. But in the nucleic acid, right, they have about extra thing. They have about the phosphorus. Okay, because they have a phosphate group. They have a phosphate group which is made from the phosphorus. So each of the biological molecule, they carry different functions. For the carbohydrates, right, they will use for the energy storage, okay, which is for the glycogen or starch, or structure component by the cellulose. Proteins, okay, the example of product include the enzyme, the defense hormones, any kind of protein. So you can see the bond, okay, carbohydrate is glycosidic bond, protein is peptide bonds, nucleic acid is a phosphodiester bond. See, it's not hydrogen bond, it is phosphodiester bond, which is the sugar phosphate group. Because they make their backbone, they can join from one nucleotide to another nucleotide, join them together. So this lipid by the ester bond, okay, see, there is the ester bond. So lipid, they have their function, okay, which is they will become the source of energy. Lipid is not a primary source of energy, just it becomes a source of energy. And then they can do the insulations, hormones, those kind of thing. Okay, so just remember condensation and hydrolysis. how it able to occur. Okay, finish. And then, okay, Neil, I think we can try some question. Just a few questions, a little bit. Okay, Neil, tell me, which is the answer for this? Number 6.
The corresponding one, yeah.
Yeah, the corresponding one. So the complement-based pairing. Okay, so how's the answer? A to which one? A to
Is it T? So is it D?
D. Okay, C, G. Yeah. It's T. Oh, donkey. Yeah. Your answer is correct. Okay, so very easy, right? Yeah, just like this. Okay, for this one, what is the X? Which one?
Base B.
Yeah, it's a base. Yeah, the nitrogenous base, the ATCG, 4 type of base. Okay, so I think this you know how to do it. Not show it. Okay, this one. Okay, which diagram show the section of DNA from the organism that is most closely related to the chimpanzee? Which one is most closely related to chimpanzee? Most closely related. So the answer is almost the same.
And A.
Yeah, it's A. Okay, because there's only one base difference. Okay, so IGCSE is very easy when you know how to answer it. Okay, so Neo, do you have time to do the homework?
Yeah.
Okay, I give you this homework. You can take your time to complete it. So next week we start learn about chapter 5. Okay, and then I'll give you this one also. You can read. Just ignore the Mandarin word. Just look at the English word. Okay, bye.
Okay, bye.
Bye.